Cancer. It is unfortunately a disease that has affected all of us, be it through our own experience, a family member, or a friend. Breast cancer alone accounts for 1 in 3 of all new female cancers annually in the US, and a common treatment for this are CDK4/6 inhibitors. However, patients sometimes develop resistance to the treatment, and it is not understood why that is. Exosomal microRNAs, being mediators of cell-cell communication, could play a key role in the regulation of treatment response and resistance. And by investigating further into exosomal microRNAs and their associations with treatment outcomes, we can further our understanding of their underlying mechanisms. As such, the purpose of our study is to determine microRNAs and their related biological pathways as predictors of CDK4/6 inhibitor outcomes in metastatic breast cancer patients. Our study had 11 patients who were being treated with CDK4/6 inhibitors, 5 of whom responded to treatment, and 6 of whom did not. We had each of them have their microRNAs analyzed with differential expression, comparing treatment resistance and response, resulting in 54 microRNAs that were associated with treatment resistance and 18 microRNAs that are associated with treatment response. This does indicate that microRNA pattern expression could play a key role in prediction of treatment outcomes. We then made a network analysis of microRNA gene-target relationships, and from there took those target genes and identified pathways that were enriched by them through an enriched pathway enrichment analysis to find pathways that were associated with our treatment outcomes. With these pathways in mind, we found a review paper that proposed several mechanisms of resistance to CDK4/6 inhibitors. While comparing these, we found that while there weren't any that were associated with treatment resistance, there were 3 that were associated with treatment response, showing that there are actually many pathways that are associated with and support the proposed mechanisms of resistance. Here we have a comparison of microRNA network instructions showing the interconnectivities of our microRNAs in the blue boxes, interconnectivity to their gene targets in the purple dots. As you can see with our treatment response on the left, there are many more connections than that of treatment resistance on the right, possibly explaining the 3 mechanisms of resistance that were exclusive to treatment response. In conclusion, our study has shown that pathway analyses of our microRNAs has supported several hypothesized mechanisms of CDK4/6 inhibitor resistance. As such, we have demonstrated the feasibility of using exosomal microRNAs as predictors of CDK4/6 inhibitor outcomes in metastatic breast cancer patients. This is huge for the field of precision medicine in particular because we can find patients who are more likely to respond to treatment better and help them manage their cancer faster. Future studies could utilize a larger patient sample size and therefore potentially more differentially expressed microRNAs for more conclusive results in the future. Thank you to all the patients who participated in this study, as well as Dr. Chen and others who helped mentor me through this project. And thank you for listening.